Hello students, in this video first I will start with viruses. R.H. Whitaker did not mention some acellular organisms like viruses, viroids in his 5 kingdom classification. So it was a drawback of his classification, but we have to study all these. So let's start one by one. First we will start with viruses. Virus is a connecting link between living and non-living. Viruses cannot be classified into living or non-living things as they have features common to both. Clear, children? Also, they have many unique features of their own. Viruses are intracellular obligate parasites. Obligate parasite, do you know, children, what is the meaning of obligate parasites? An obligate parasite is a parasitic organism that cannot complete its life cycle without exploiting a suitable host. It means if an obligate parasite cannot obtain a host, it will fail to reproduce. So, viruses are intracellular obligate parasites which lack cellular organelles like mitochondria, ribosomes, etc. Viruses do not have cell-like arrangement. So here they act as non-living. Chemically, virus is a nucleoprotein. It means outer helical structure. You can see in the figure, this is helical structure. It is made up of protein. It is a coat or covering which is known as protein capsid. And inside this capsid, what is present there? Nucleic acid. Either it will be DNA or RNA. Both cannot be present together. Clear, children? Either it will be DNA or RNA. So it is a nucleoprotein because outer protein layer is present which is known as capsid, and inside the capsid nucleic acid is present. Therefore it is a nucleoprotein particle. They are non-cellular organisms because there is no cellular structure. They are characterized by having an inert crystalline structure outside the living cell. If they are present inside the host cell, then it behave like a living. But if it is outside of the living cell, then it is present in a crystalline form. Clear, children? One thing you always keep in your mind, that virus always needs a host cell to multiply. Is it clear? If it is outside of the cell, then it is only nucleoprotein particle. Like non-living crystals, it can be crystallized like any chemical and stored in bottles like sugar cubes or crystals, etc. for a long time. And if it is present in host cells, then it will damage the cells and destroy all DNA. When virus attack a host cell, it controls the host cell's machinery, and then it will replicate here and kill the host cells, kill the DNA. So when it is present inside the cell, host cell, then it behaves like a living. The term virus, very important line, the term virus, that means venom or poisonous fluid, was given by Pasteur. So there are some features in virus due to which we can say that it is living. But if it is present inside the host cell, what are these features? That viruses have genetic material, DNA or RNA. They mutate. They can grow if present inside the host cells. They can be transmitted from one host to another. They are capable of multiplication within a host. They react to heat, radiation and chemicals. They show irritability. They bring about enzymatic changes in vitro. They are able to infect and cause diseases to living beings. The DNA and proteins of viruses are similar in composition and structure to those of higher organisms. So these are the features by which we can say that viruses are living. They behave like non-living also. How? They can be crystallized like an ordinary chemical and stored in the bottle. Outside the host, viruses are inert. Inert means lacking the ability or strength to move. There is no cell wall, membrane or cytoplasm. There are no cell organelles and there is no metabolism. They do not have functional autonomy, that is, they are not capable of any functions unless they obtain metabolic products from others. Clear, children? And energy producing enzyme system is absent. If virus will attack on healthy cell, suppose there is a healthy cell, if virus will attack on this cell or living cell, then it will inject only nucleic acid, DNA or RNA. Clear? It will inject only nucleic acid. Whole virus never get inserted. Whole virus never get inserted in the healthy cell. Clear, children? Only nucleic acid, this one will insert inside the host cell. So when nucleic acid will enter inside the cell, it will capture the cell completely. It will destroy the DNA which is already present inside the cell and will produce protein for itself. Clear, children? So these are the characteristics of living. Now we will discuss about some important milestones in the history of viruses. A Russian biologist, D.J. Ivanovsky, studied the diseased tobacco plants in 1892. The diseased plants had mosaic pattern of light green and yellow areas on the leaves. Students, you have already seen mosaic pattern in some houses. Floors are in mosaic pattern. So same way, the diseased plants had mosaic pattern of light green and yellow areas on the leaves. Ivanovsky prepared the extract of infected leaves. He took that infected leaves and prepared the extract and passed it through a filter that could trap all bacteria. The filtered fluid did not contain any bacteria, but to his surprise it was still highly infectious. It caused the same mosaic pattern when sprayed on the healthy leaves. So he recognized that microbes as causal organisms of the mosaic disease of tobacco plant. Clear? So the credit of discovery of viruses goes to D.J. Ivanovsky. Ivanovsky recognized only microbes. He did not give name. About 6 years later, a Dutch botanist, M.W. Bizarinek, in 1898 repeated the experiment and gave the term virus. Clear, children? He only gave the term virus, meaning of that word venom or poisonous fluid, to the infectious agent present in the infected sap. Tobacco mosaic virus was the first virus to be discovered. In 1935, W. M. Stanley isolated the virus in crystalline form. It is very important class, so learn it. In 1935, W. M. Stanley isolated the virus in crystalline form for the first time. In 1946, he performed electron microscopic study of a plant virus. Viruses cause diseases like mumps, smallpox, herpes, and influenza and AIDS. In plants, the symptoms of virus can be mosaic formation, leaf rolling, curling, yellowing, and vein clearing, dwarfing, and stunted growth. Now we will discuss about our next topic, structure of tobacco mosaic virus. Already I have informed you that tobacco mosaic virus was the first virus which was discovered in 1892 by D.J. Ivanovsky. This is the image of leaf of tobacco plant. The first visible symptoms of tobacco mosaic virus infection in tobacco plant appear in the form of downward curling and distortion of young apical leaves. The leaves show typical yellow and green mosaic symptoms. The electron microscopic studies have revealed that tobacco mosaic virus has two constituents, a protein coat which is also known as capsid and nucleic acid. Students, already we have discussed, this one is protein capsid. Is it clear? And inside that nucleic acid is present. This capsid is this one capsid. This is made up of identical protein subunits which is known as capsomeres. You can see this one. Clear? This is the subunit. And this subunit are helically arranged around a central stranded RNA molecule. You can see this. Clear, children? This protein coat is capsid is made up of subunits which is known as capsomeres, which are helically arranged around a central stranded RNA molecule. The genetic information which is necessary for the formation of a complete tobacco mosaic virus particle is contained in its RNA. Clear, children? The complete information, genetic information which is necessary for the formation of tobacco mosaic virus particle is contained in its RNA. It is possible to remove the protein coat from RNA, and the naked RNA is capable of infecting tobacco plant. Already I have informed that whole virus do not insert inside the host cell. Only nucleic acid enter inside the host cell. Once it enters inside the host cell, the RNA directs the protein synthesizing apparatus of the host cell to synthesize its own proteins. Clear? So, RNA has two functions. First one is self-replication and the second one is synthesis of virus specific proteins, for which it takes raw material from the host cell. Now the next one is bacteriophages. Viruses which infect bacterial cells are known as bacteriophages or simply phages. Bacteriophages were discovered by Edward Todt in 1915 and D. Harrell in 1917 independently while investigating certain types of soil bacteria. Let's see the structure of bacteriophage. You can see in the figure it looks like tadpole with hexagonal head. You can see this is the head part. Hexagonal 1, 2, 3, 4, 5, 6. So hexagonal head and a cylindrical tail. When we see the structure of head part, head consists of a tightly packed core of double-stranded DNA. This is double-stranded DNA which is surrounded by protein coat or capsid. Already I have explained. The tail composed of this tail part composed of contractile sheet surrounding the hollow core. This is terminal base plate and these are tail fibers. So tail, you can see that tail fibers are attached with terminal base plate. Now viroids. We will discuss about viroids. Viroids are small single-stranded circular RNA agents which infect plants. They are the smallest known pathogens and are naked, circular, single-stranded RNA molecules that do not encode protein. There is no covering of protein. The discovery of first viroid goes back to 1967 when Diener and Raymer reported that the agent of transmission in the potato spindle tuber disease was a naked RNA and it has no nucleoprotein covering. So, T.O. Diener in 1971 introduced the term viroid